Welcome everyone. Thanks for coming to the second caregiver training for our parents group and also a community chat meeting. We're combined today with some folks from our community that don't usually attend the parents meeting because we had a lot of questions from people about the drug development process, where we are in that process, how we can impact change, and what we can expect with some of these treatments moving closer towards clinical trials. So, my name's Heidi Wallace. I'm with Association for Creatine Deficiencies, if I don't know you, and I am leading this project with Audrey Thurm, who is on with us as well. She's a clinical psychologist at the National Institutes of Health and was part of the Vigilance study for CTD, so has a really strong background in our disease space and was part of the Last Parents Project. And I also have Emily Reinhardt, who is working on this project, and she's our registry coordinator. So if you haven't joined the registry, she's the person to talk to, or if you're locked out, or if you have any questions, contact Emily. And then last, Celeste is on the line, and she is the trainer, the parent trainer in this project, and she'll give you her background here in just a minute. But I'm going to just do some housekeeping items and say that please go ahead and throw things in the chat during the meeting, and we'll be taking little breaks every few slides and checking the chat and asking you live if you have questions, so that if any of this is new to you and you want to learn more midstream, don't be shy to ask. And this will be, like I said, recorded and then posted, so don't feel like you have to take a ton of notes. Just enjoy the meeting, and we'll have this up in just a few days. So a few ACD updates. Right now is our Holiday Heroes campaign. This is a special campaign every year because every dollar that we raise is reserved for research. And it really quite literally determines how much research we can fund and drive and encourage in the upcoming year. And so you probably noticed every year we're kind of raising our goal a little more. Last year we raised over $200,000, and this year we're shooting for $300,000 because we have a lot of projects to support. But we do have one awesome individual who said, you know what, if your community can rally together and raise 200,000, I'm going to donate 125 and put you over the top. So like no other year, this year is the year to really consider helping out. And I've got some ideas of how you can do that. Some things that we are looking forward to that we can talk about at this point is we have our fellowship applications in. There's some really awesome, really groundbreaking new work that people want to start in our space that we hope to support. We're also going to be continuing supporting the drug repurposing project at Dr. Stockler's lab, and we should have results from that by the spring. As a reminder, this started with a fellowship project that we funded where they identified some drugs that are already approved for use in other disorders that could be potentially used in CTD to get creatine into the cell. So they're checking this in human cells now to see if it pans out. And we also have a creatine uptake screening going on at the University of Utah at our Creatine Deficiency Research Center, and there will be more work on that as well. Those are just a few things. There's a lot cooking in the background, I will say. So how can you help out? You can set up a fundraiser if you haven't already. Contact us, go to our website, to Holiday Heroes, email us, text me anytime. I will help you personally set up your fundraiser. You can make a donation that is meaningful to your family. You can share our posts that are coming out regularly, forward an email that you find inspiring. We have, hopefully everyone's getting the emails. If you're not, there's a whole series right now of stories about featured patients in our community, and they're really sweet and touching, and I think those are great to forward to friends and family. You can give a donation on behalf of someone that you care about. We have thank you cards, and a donation was made on your behalf that you can send in your Christmas cards or whatever holidays you celebrate. This on the right is an example of a Roundup campaign. It's a bar in the Portland area that is featuring our kiddos on their coasters and on little signs, and they've got the symptoms and they've got the research that we're aiming to fund. And this is the second year they've done this. Last year they had no materials, no explanation, and they just asked people for the last 2 weeks of the year, do you want to round up for this charity you've never heard of? And they said yes, 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 and we got about $400 from doing that. So this year we're campaigning hard with this partner, providing them these materials that cost us very little, and I think it's going to be amazing. So if you have a friend that runs a cafe or a bar or anything like this where you think this is a small ask, it doesn't cost them anything to just share the word, let us know. We will help you get it set up. So live on location, February 1st, we have some big announcements that we're really excited to share, and we're going to do some Facebook Lives, and so just get ready, February 1st. I can't tell you anymore. Community chat series. Upcoming is January 3rd, our first ever CCDS Dads Chat. So dads that are on, join us. And we want you to have a strong network too. Moms, invite dads. Hopefully we'll get a good support group going. February 7th, we'll be discussing some of those announcements that were made on CCDS Day on the 1st, and some exciting opportunities for all of us to be able to participate in research in 2025. We will have guest speakers that will be presenting this research, so it'll be a really important meeting to attend. And then March 7th, we have guest speakers again talking about how creatine deficiencies are inherited, including things like mosaicism and diagnosis, especially diagnosing females with CTD. We aim to answer the many questions that are floating about and record that meeting, so we have a great resource for future parents. Walk for Strength, number 10, our 10th Walk for Strength is June 21st, 2025. So put that on your calendar, and we'll start the whole process of signing up teams and getting going after CCDS Day, mid-February. And finally, for our parents who are on the call from the 2.0 group, the Research Jam, everything I heard was fantastic, that our community is like the best group of families that they have worked with, and they were so excited about the conversations, and I heard some great things from a lot of you. So hopefully all of that is coming together in their report that we will have by February. So we'll have some great data, and that's your participation in those meetings is going to drive this whole project. And as other groups meet, like our subject matter experts, which we have now formed, we're going to be referring to what you said as we talk about what's important in our community and what changes are meaningful. So the expert groups that we have launched, we have a group that is talking about spectroscopy and how we can have better guidelines for using MRS in our patients to have a quantitative measure of creatine. So we hope that that's going to be a really positive impact in research for creatine deficiencies. Plasma GAA is another group, and then EEGs as a measure of seizure activity. There are mentors in our group. I know we've only met once now because of Research Jam last month, but as a reminder, everybody will have a mentor. One of our parents that were involved in the first group are now mentors in this group, and they'll be emailing you and having an interview to talk with you about setting up your presentation that you'll be presenting over the next 2 years. So all of that is kicking off soon, and I just wanted to give you a reminder in case you get a strange email from someone that's what that's all about. And our next meeting is January 10th. And that should be on your calendar, so please let me know if it's not. And now I'm going to turn it over to Celeste to tell us all about drug development.
Awesome. Hey guys. So this is just a brief agenda for today. We're going to talk about the drug development process, as well as outcomes and endpoints, and then we will have a Q&A at the end. Next slide. All right, so I am Celeste. First and foremost, I'm a mom. I have 4 kiddos. My oldest has CTD. We're in North Carolina. I am the director of education for the ACD, and I'm also the working on this grant as the parent trainer. And so my background's in special education, and I did a lot of professional development or led a lot of professional development. So this is kind of right up my alley. I do just want to give a disclaimer that all of this information was gleaned from NORD's Rare Disease Drug Development Series, What Patients and Advocates Need to Know, as well as a PowerPoint that Audrey Thurm developed called Clinical Trial Assessment Terms to Know for Caregivers. This is going to be really information heavy, but we are going to post it and or email it out. So just kind of hang in there with us. Next slide. All right. So as we get started talking, looking at international drug development, there are many regulatory bodies around the world that approve drugs in their region or country beyond what's listed here. This is just a sampling. So you have Canada, Health Canada, US, the FDA. In Europe, it's the EMA, European Medicines Agency, Africa, African Medicines Agency, et cetera. So today we're going to be talking about the focus on the FDA drug process in the US. However, this is very relevant to all of these regulatory bodies, as many will more quickly work to approve an FDA vetted and approved drug, and many have very similar regulatory processes. So next slide. All right. So there are over 7,000 rare diseases. And with the rare diseases, there's always usually a poor understanding about them, including the different variations within the disease. There are limitations in funding and researchers. And then there's a small number of patients, within most rare diseases who are geographically dispersed. We have families all over the world, as do a lot of rare diseases, and more than half the patients with rare diseases are children. When we talk about an orphan drug is a drug or biologic that treats rare diseases. And so that leads me into talking about the Orphan Drug Act of 1983. This was enacted to give incentives to invest in development of treatments for rare diseases. So some of the incentives include market exclusivity, where there's 7 years of market exclusivity for the drug, tax credits of 25% for qualified expenses that are incurred during clinical trials, exemption from fees like marketing application fees, the FDA marketing application fees. Then there's grants, so federal research grants for clinical study of new tests or therapies to diagnose and treat rare diseases. To be eligible for the benefits of the Orphan Drug Act, the drug has to be developed to treat a rare disease with fewer than 200,000 people in the US. There's no reasonable expectation that the drug's development costs will be recovered through sales of the drug. And we just want to make a note that all 3 creatine deficiencies qualify for orphan drug designation. Next slide. So there are 5 steps in the drug development process, and we're going to dive into each one a little bit deeper in the slides following. This is just kind of an overview. Next slide. All right, so the first stage is preclinical research, looking at discovery. So this is an exploratory phase where you're going to be doing some research looking at the basic biology of a disease. And then there's going to be lots of laboratory studies to discover a potential treatment and to better understand what that might look like. So this is the very, very beginning. Next slide. So as we think about creatine deficiencies in this preclinical research discovery phase, this slide is going to show several CCDS specific projects in the preclinical research discovery phase, many who have received fellowships thanks to Holiday Hero funding. Just want to give a shameless plug there. So Dr. Schlabach with his in silico modeling of CTE compounds, Dr. Filippo Angolia, looking at creatine uptake, lip shoots, looking at organoids and gene therapy, Edwin looking at small molecules to deliver creatine across the blood-brain barrier. Leon looking at nanoparticles, doing the same thing. Baroncelli looking at gene therapy, as well as Dr. Bressant and Dr. Walia. So this kind of just gives you an idea of all the projects that are in the discovery phase right now. Next slide. All right, so as we move on to preclinical research in the development phase, this is where we're going to look at a few different things. So this is where safety and toxicity are considered. Is it safe to give humans? Dosing, how much is tolerable of a therapeutic and accomplishes the intended goal without giving too much? Route of administration, what is the best route for a drug or therapy? So you're looking at IV, a pill, an injection, things like that. And all of this is going to lay the groundwork for clinical trials. And then once all of this is completed, then we look at filing an investigational new drug application to the FDA for approval. This includes information about the drug, how it's manufactured, a proposed study protocol, and the FDA is going to provide feedback on this application within 30 days once it's submitted. So looking at preclinical research development, this slide is going to show several CCS specific projects in this phase. So Dr. Walia, again, with his gene therapy toxicity, Dr. Lipschutz with gene therapy manufacturing, and then Ultragenyx and Sarah's. Sarah's having a nasal spray nearing phase 1, and Ultragenyx small molecule. So this is super exciting to see all of this kind of written down in one place. Next slide. So as we look towards clinical trial readiness, the ACD has also done a lot looking towards patient-focused trial design. So we have our creatine info natural history study. If you have any questions, Emily put her email in the chat. And then FDA awareness. So we have done an externally led patient-focused drug development meeting, which resulted in the Voice of the Patient report. And then Heidi presented at a CBER. We'll go over what that is, but that's a center within the FDA last week, I believe. We've, through our first parents grant, developed a core outcome set for creatine deficiencies. In this second PCORI grant, we're working on outcome measurement tools considerations. And then we're working on and supporting developing biomarkers through FNIRS. In conjunction with Boston Children's, and then also organoid studies through some of our fellowships. So this is super exciting and we're moving mountains. So I just want to take a second right here and just pause. If anybody has any questions on what we've talked about so far, you can unmute or put in the chat. Okay. All right, we'll keep going. So next slide. So as we move into the next phase, this is going to be clinical research. So clinical trials consist of 3 phases before a sponsor applies to go to market with the FDA. Phase 1 is going to be looking at safety and dosage. This is where they're going to test the therapeutic with a small number of healthy trial participants. And you're going to continue looking at and determining the safety of the treatment. Phase 2 is going to be looking at safety and efficacy. So this is going to be testing of the therapeutic with a small number of affected trial participants. So like those who have CTD or who have GAMT. And it's going to continue to determine the safety of the treatment as well as the effectiveness. And then as we get to phase 3, it expands. So you're continuing to look at efficacy, and then you're also looking at side effects. So this looks at testing of the therapeutic is expanded to a larger number of affected trial participants, and it further establishes safety and effectiveness of the treatment. Next slide. Super exciting. This slide shows a current clinical trial that some of our kiddos are participating in, and hopefully it's just the beginning of our involvement in clinical trials. But Dr. Greenspan's testing of phenylbutyrate in patients with seizures. Next slide. All right, so as we move towards the FDA approval, so a new drug application or an NDA or a biologic license application, a BLA, those are after clinical trial, the drug sponsors would submit a marketing application, which is what those are called. And it depends on, so for example, if it's a small molecule drug, it would be an NDA. If it were a gene therapy or a cell therapy, something like that, it would likely be a BLA. And so after the trial is completed, the drug sponsors are going to submit that marketing application to the FDA. Once the FDA gets it, they have 60 days to complete a file review, which means they're going to make sure that they have all that they need once the application is submitted completely. And then the waiting ensues. They'll review within 10 months. And the application is assigned to a review team and possible advisory committee that includes providers, patients, pharma, researchers, and they're going to review all the information and inspect the manufacturing. They're going to look at the benefits versus the risks, and they're going to make a decision on whether they're going to approve the drug or therapeutic or deny it. If additional input is needed, the drug sponsor can resubmit the application and it goes into a review cycle. So they can add additional data or collect additional data and then resubmit, and it just goes into a cycle. I do want to take note of the Prescription Drug User Fee Act, or PDUFA. This was enacted in 1992. It allows the FDA to collect fees from drug manufacturers to help fund the drug approval process. And it aims to speed up the drug approval process while ensuring the safety and efficacy of new drugs. So before we continue again, I just want to pause for questions if there are any. All right. Next slide. So as we talk about the FDA, I mentioned this earlier, there are 4 centers housed under the FDA. So CEDAR is the Center for Drug Evaluation and Research. And some examples that you might find at this center would be drugs, some biologic therapeutics, some things you wouldn't consider drugs like fluoride toothpaste and sunscreen. The next one is CBER, so the Center for Biologics Evaluation and Research. And some examples that would fall under this center would be vaccines, blood or blood products, cell, gene, or tissue therapies. The next one is this CDRH, the Center for Devices and Radiological Health. Some examples here would be medical devices or radiation emitting electronic products. And then CFSAN would be the Center for Food Safety and Applied Nutrition. And this includes foods, including medical foods, dietary supplements, and cosmetics. Next slide. All right, so FDA drug review programs. They were established to support the rare disease drug development process, and they're intended to facilitate and expedite development and review of new drugs, to address unmet medical needs in rare diseases. They do still require drug sponsors to gather and provide compelling evidence in support of a drug's safety and efficacy, which does take time. So the first one is a breakthrough therapy designation. This provides additional opportunities to engage with the FDA early in the drug development process, which will help strengthen and improve the clinical trial study design. It's used when early studies in humans indicate that the drug being studied may be substantially better than what is currently available. The next one is a fast track designation. The marketing application receives a rolling review. So that means that they're going to review each section as the application is completed and submitted, and as well as other benefits. It's used for drugs intended to treat serious conditions and fill an unmet medical need. So like conditions that have a large impact on daily life and will get worse with no treatment. The next one is accelerated approval. This allows the possibility of approval based on a surrogate endpoint or intermediate clinical endpoint instead of a traditional clinical endpoint. And I know those are some big terms. We're going to talk about those in a little bit. It does still require that the sponsor conduct a trial on the traditional clinical endpoint to confirm that the drug has clinical benefit. This is used for serious conditions with no or few treatments. The last one is a priority review voucher. The FDA agrees to act on a drug sponsor application within 6 months rather than the 10 months required under the standard review. So, 4 months can make a huge difference. It's used for drugs that would provide significant improvements in the safety and effectiveness of treatment, diagnosis, or prevention of serious conditions compared to standard applications. I do want to take note of there is a rare pediatric priority review voucher. This disease program aims to incentivize drug development for rare pediatric diseases. Under this voucher program, a sponsor who receives an approval for a drug or biologic product for a rare pediatric disease may qualify for a voucher that can be redeemed to receive priority review for a different product. The sponsor could also transfer or sell the voucher to another sponsor. Sarah has announced last month that they have a rare pediatric priority review voucher. That's an example. Next slide. All right, special provisions. Expanded access and compassionate use is the first one. This is access to investigational drugs outside of a clinical trial when there's a serious illness or there's not a therapeutic available that's similar or satisfactory, and when all other treatment options have been exhausted. So, for example, a trial not enrolling nearby or that's close to new participants or a patient doesn't meet the inclusion or exclusion criteria, the FDA has 30 days to approve this provision unless immediate access is authorized due to an emergency. So if the FDA approves it and the sponsor says yes, a provider will need to be identified who's going to agree to a few things. They're going to need to oversee and report on the progress as well as adverse reactions. They're going to need to work with the FDA, the drug sponsor, and the IRB, which is I believe it's Internal Review Board. And then they're going to need to complete and submit an annual report and submit a summary of the patient experience after therapy is complete. There are different kinds. So for example, like a single patient investigational new drug, like an N of one, so a trial with one patient, a single patient protocol, or emergency expanded access. And then the other special provision is drug repurposing. There are over 20,000 FDA approved drugs. So a supplemental new drug application is submitted by the original drug sponsor and the new drug sponsor. And this is still shorter than a clinical trial. There needs to be knowledge of the disease, especially on a molecular level, and providers can and do try off-label use. Next slide. So this slide shows a CCDS specific project looking at drug repurposing. Again, a recipient of a fellowship thanks to Holiday Hero funding. Another shameless plug. All right, so additional measurement terms. Statistical significance. When I was preparing for this presentation, I realized I'm not a statistician. So statistical significance is a mathematical measure of the difference between groups. It is statistically significant if it's greater than what might be expected to happen by chance alone. It's often represented by a p-value, and the range is 0 to 1. The smaller values are more indicative of significance. The FDA usually requires a statistically significant treatment effect to approve an intervention, but it isn't the only criterion. It can be related to sample size, so even the smallest effect could be made significant with a large enough sample. And then another measurement term is clinical meaningfulness. This is the patient or caregiver's judgment of the importance of a concept or treatment effect. FDA approval depends on statistical significance of a treatment effect, but also the clinical meaningfulness of that effect. For treatment effects, clinical meaningfulness is a personal evaluation of the risk-benefit ratio, and it can be evaluated through qualitative and quantitative methods. Next slide. All right, and the last phase is post-market safety monitoring. And this is just the FDA continues to monitor for safety after approval of a drug or therapeutic, both in the manufacturing and consumption of it. And so now I'm going to take another quick pause to see if there's any question. All right, we'll keep going. All right, so we're going to kind of flip, and now we're going to talk about endpoints and outcomes. So an endpoint is a specific, clearly defined variable that is being measured in a clinical trial. There are 3 different types. So a primary endpoint, that's a main piece of data that's evaluated for FDA approval. A secondary endpoint provides supportive information about a therapeutic's effect on the primary endpoint or demonstrates additional effects on the disease. And then an exploratory endpoint may include clinically important events that are less likely to occur, but can explore new hypotheses. So as we think about that, an intermediate clinical endpoint is a measure of therapeutic effect that is considered a reasonable predictor of clinical benefit that occurs later in the progression of a disease than the intermediate clinical endpoint. This provides more direct evidence and therapeutic effect on the disease than a surrogate endpoint. And then lastly, a surrogate endpoint is a substitute for a direct measure of how a patient feels, functions, or survives. It is not a measure of clinical benefit, but it's likely to predict clinical benefit. All right, so that was endpoints, and now outcomes. And a clinical outcome is a measurable change in symptoms, overall health, ability to function, or quality of life. These can also measure the success of care as well as a person's response to an already approved treatment. A clinical outcome assessment measures how a patient feels, functions, or survives. And there's 4 different types. And so we're going to go into those over the next few slides. Next slide. Okay, so the first one is a performance outcome. This is a measurement based on standardized tests or tasks. So for example, a walk test or the Stanford Binet intelligence scales. It's often administered by someone with specific training. So for example, a psychologist would administer a cognitive test. And then sometimes it can be administered by a patient themselves. So for example, a timed up and go test. You are asking a patient to perform tasks in this outcome measurement. And so it does require patient cooperation and motivation. Next slide. And the next one is a patient-reported outcome. This measurement is based on a report that comes directly from the patient about the status of their health condition. It's without amendment or interpretation of their response by anyone else. It can also assess the patient perspective on functioning that it could be observable by others. It's measured by self-report or by interview or rating scales. And it's the only way to measure symptoms or unobservable concepts that are known only to the patient. Next slide. Clinician reported outcome. This is a measurement based on a report that comes from a trained healthcare professional after observation of a patient's health condition. It requires the knowledge and or judgment of a healthcare professional to be interpreted and reported. It includes things like clinically observable signs, behaviors, clinical manifestations, biomarkers, clinical findings, and events. And the last one, we likely have all participated in this type of outcome, the observer reported outcome. And this is a measurement based on a report of observable signs, events, or behavior related to a patient's health condition by someone other than the patient or healthcare professional. It's usually reported by a caregiver. So for example, you may see rating scales, interview, data collection, etc. It is particularly useful for patients who can't report for themselves. So young kids or individuals with intellectual developmental disabilities or with speech delays, things like that. All right, and reliability and validity. So I hope this visual is as helpful to you as it was to me, but just kind of honing in on these terms. Reliability is consistency or dependability of a measurement. It's determined by doing the same test multiple times under the same conditions, and you get the same results. And then validity is looking at the accuracy of a measurement. It's crucial because you need to be measuring the correct effect of a therapeutic. It's measured by comparing results of a test or therapy against a known standard. Tools used must be calibrated to ensure the results they produce are valid. And the results obtained from calibrated tools can be used to confirm the validity results from non-calibrated tools. And then the last term is responsiveness. And this is just the ability of a tool to capture meaningful change. And so with that, I just want to pause one more time for questions before we watch a quick video. All right. So clinical outcome assessments and endpoints, sometimes these terms are used interchangeably, but they do have distinct meanings and uses. So we're going to watch this video from NORD's Rare Disease Drug Development Series. It's called Understanding Clinical Outcome Assessments and Endpoints. We're going to stop the recording, but we do have the QR code available for you guys to register for the course. It's free and watch it as well on your own time. So slides and just a couple more slides and then we'll be finished for the evening. So we just wanted to point out that we have additional resources for clinical trial readiness and the drug development process on our website. Um, and our website has been revamped, or is in the process of continuing to be revamped. So it's not going to look like what the slides look like because it's recently happened. However, if you go to creatinginfo.org/resources or /drug development, you'll find some additional resources. And this presentation will also be housed either there or on our creatinginfo.org /parent staff project page. So those are just a couple different places where you can find those resources. And now we just have some space for Q&A. Audrey Thurm is on here to help answer some questions as well. We had a couple good questions earlier, but what questions do you guys have related to all of this information? I know it's a lot to process, especially on a Friday evening. Anything? All right. Well, if nobody has any questions, I won't belabor. But the last slide just says Happy Holidays, Happy New Year. And the Parents 2.0 participants, will see you Friday, January 10th, 2025. And then for the community chat series, we'll see that one is for the dads in January. So the dads will meet up with Dan and Randy on Friday, January 3rd. And with that, you guys have a great evening.